Hello, and welcome to Lecture 2-2, where we're going to be talking about the Psychodinae, the moth and sand flies. These guys fall under the Diptera, which are flies in general, and of course the Diptera of medical importance. A little bit of life history on flies. Remember, in insects, some insects are what we call holometabolists. Most flies are. They come from an egg. The egg will hatch into larvae, then pupa, then adults. Adults that are egg-laying, we have different types of egg-laying that occurs even within the Diptera themselves. Oviparous organisms will lay eggs that will hatch after they've been laid. Ovoviviparous are eggs that that are fertilized, they develop as eggs, and then the egg hatches within the female and then is laid as a live organism, a larva. And then larviparous is just live birthing in general. Hematophagous flies are blood feeding in both the, and most of the sand flies, of course, are blood feeders. When we have flies that are blood feeders, some insects are what we call autogenous and others are anautogenous. In autogenous organisms, these insects don't require a blood meal for survival or egg development, but usually their life cycles are very short. Think mosquitoes in Alaska sort of thing. And the feeding time, the amount of time they have for feeding is it's a very short window. So they're going to get the richest meal they possibly can. And in this case, it's going to be blood. Anautogenous insects are those in which their eggs will not develop unless a blood meal is given. It's so nutrient dense and they need so much of the nutrients and iron and protein found in red blood cells that it's actually required for their young to develop even in the egg. So in classification of Diptera, we have two suborders. We're going to take a look at the Nematoceras and the Brachycera. Today we're looking strictly within the family Psychodidae in the Nematocera. These are, we'll look at a few moth flies in that too, but we'll move into some other lectures to get into the black flies, horse flies, those sorts of things, and midges. Mosquitoes, the Culicides, we have an entire unit on those. So in the Nematocera, I can't talk about the Nematocera without mentioning mosquitoes and the Culicidae family. They are the most prolific of all vectors. They spread all kinds of different diseases, but we're going to spend an entire unit looking at mosquitoes, so I don't want to spend time on those now. What we're going to be looking at are primarily these sand flies. And sand flies cause a number of different diseases, and some of them are really quite awful. You can see here, this is a form of Leishmaniasis, and they have some really interesting 2- and 3rd-party type life cycles. So we'll take a look at that as well. So here we're going to watch a quick video. This is a scientist who actually is researching sand flies, and he's going to dissect the gut of a sand fly so we can see what is inside of it. Oh, sorry about that. That's a software issue I have.
Hello, my name is Hector Diaz Albater, and I'm a scientist working on sandflies at the University of Glasgow. Sandflies are blood-feeding insects similar to mosquitoes and midges. They transmit Leishmaniasis, a nasty parasitic disease similar to malaria that affects humans, dogs, and other mammals. The disease causes painful and disfiguring lesions. It can also affect the liver and spleen and become fatal. 12 million people are currently infected with Leishmaniasis, and 350 million in 88 countries around the world are at risk. Leishmaniasis is found mainly in tropical and subtropical regions. However, global warming is changing the disease distribution, and now we have more cases in southern Europe. Currently, there is no vaccine, and the drugs used for treatment are highly toxic and can cause serious side effects. Transmission takes place when a healthy sandfly takes a blood meal from an individual infected with Leishmania and ingests the parasites. Inside the sandfly, the Leishmania parasites develop and replicate exclusively in the gut into new infective forms. This infected sandfly will then transmit the disease when it next feeds on a healthy individual. injecting the parasites and infecting them with Leishmaniasis. This is one of the reasons why we scientists are very interested in sandfly guts. We use this tissue to perform experiments to further understand the parasite life cycle and to develop new treatments. In this video, I will show you how to dissect a sandfly gut. First, I place the flies in cold dissection solution for a few minutes to anesthetize them and keep everything on ice until needed. Then, I take a microscope slide, place it on the microscope at a drop of dissection solution, and then using the plastic Pasteur pipette, I transfer the fly onto the slide like so. Before we start, please note the size of the sandfly. It's tiny. It's about 2 millimeters long. Now imagine trying to perform surgery on an animal this size. Once the fly is on the slide, use dissection needles to set it sideways. Put its posterior facing towards your dominant hand. Then, using your needle, cut the last 2 segments of the abdomen. This fly fed on blood 2 days ago, so her eggs are starting to develop. Carefully push the eggs out and move them away. At this moment, the gut is still attached to the head. Cut the head off and then proceed to push out the gut with the side of your needle, as if you were squeezing out toothpaste. Once the midgut is free, use your needle to transfer it to wherever you need. I place mine in the small tubes. Once we have collected the sandfly gut, we can perform different experiments with this tissue. Some researchers do this with wild-caught sandflies and look for Leishmania parasites. Thanks for watching. For more information, log on to the websites on the screen and follow us on Twitter. Until next time.
So as you can see, there is a lot of research going on with the sandflies, and they'll use all of those midguts, of course, to take a look at the Leishmania parasite. So in the Nematocera that we're going to be looking at the two subfamilies Psychodinae, which are your moth flies, and there's a few of them that are actually hematophagous. And then we're really going to be spending our time looking at the Phlebotominae, which are the sandflies themselves. And that guy down there on the right, that's a sandfly. So in the Psychodinaes, in these moth flies, one that is of some importance, significant, is the drain fly. Drain flies are found around standing water, drains, of course. That's why they're called a drain fly. They're a major nuisance in sewage treatment and water treatment facilities, anywhere that we have any kind of sewage occurring. Sometimes they can be found in large populations in the backyards of houses where there is septic tank damage, or where there is some kind of sewage drainage into freshwater lakes and canals, those sorts of things. They are not hematophagous. However, they do transmit pathogens because of their association with raw sewage. So if someone has a broken septic tank and septic sewage is leaking into their yard, or water around the area is contaminated with sewage, then you're gonna see a lot of these drain flies, and they spend a lot of time on that sewage, and then they turn around and, sorry about that. Then they turn around and they're near people, and they get on food and other items, and they can mechanically transmit diseases that way. With really heavy infestations, these are very tiny flies, and their wings, when they die and fall apart, they cause dust and a lot of wing debris, and this can cause asthmatics to have irritations. And so there has been a single report of what's known as urinary tract myiasis, and this is because of somebody that they actually had flies growing in the urinary tract because they used poor hygiene practices. Infestations are really hard to get rid of. Really, the best way to do it is to clean up the area as much as possible. In order to get rid of these drain flies, you got to get rid of the sewage and poor hygienic practices, etc. These guys, by the way, these little drain flies, these are what you're seeing when you go to the concerts and that, and they have the porta potties. If there's a bunch of flies in it, the porta potties after a couple of days, those are these drain flies. Into the Phlebotominae, these are the sand flies. They are of the genus Litzomia. There's a New World sand fly and an Old World sand fly, and the Old World are the Phlebotomus. The forest inhabitants, they don't cause nearly as much disease as the desert inhabitants. The desert inhabitants are what are associated with the Leishmaniasis. Eggs are laid in large batches, and they'll hatch anywhere between 4 and 20 days, depending on the specific species. Larval pupation takes up to 2 months. Pupation itself is 7 to 8 days, and adults will live for a few weeks. Females are blood-feeding, males are not. The very first time a female lays eggs, she's not going to require any kind of blood meal, but any second, third, or fourth batches of eggs is going to require a blood meal. And again, this is going to be species specific. So in the New World sandfly, these are found in the Western Hemisphere, what's referred to as New World. They're found mainly in forested areas. They feed on mammals, birds, and bats. Here on the right is the L. longipalpus, and they are vectors for what's called vesicular stomatitis virus, VSV, and Carrions disease. Bartonella bacilliformis is a bacterial disease, and of course the arbovirus, VSV. We'll talk about those a little bit. In the Old World sandfly, we're looking at the Phlebotomus, and they cause Old World Leishmaniasis. They're found on the African continent, India, and Asia, and they will cause sandfly fever and Carrions disease, in addition to Old World Leishmaniasis. Sandfly fever is also known as Phlebotomus fever and Papatachi, and Carrions disease is Arroyo fever, or also known as Bartonellosis. Another parasite disease, bacterial disease. So here we have Leishmaniasis, and Leishmaniasis occurs in 3 different forms: cutaneous, mucocutaneous, and visceral. Cutaneous is what most people are used to seeing, but visceral is actually the more common form. The Sarcomastogophora phylum is what the Protistophyllum. So the Sarcomastogophora, they are known as the kinetoplastids. That's the actual parasite that causes the Leishmaniasis that these sandflies are vectors for. In New World Leishmaniasis, we have cutaneous and visceral. In Old World Leishmaniasis, we see mostly cutaneous and visceral as well. Both places might have a little bit of mucocutaneous. Mucocutaneous is the least vicious of the two, or I'm sorry, of the 3, really causes just skin lesions. The other 2 cause, cutaneous causes lesions and ulcers, very painful. That's what you see in the top right there. In the bottom right, what you're looking at is mucocutaneous. And then in visceral, this is a young child that's getting prepared for surgery. With visceral, it starts out with ulcers, but eventually it starts to infect organs, and it becomes multiple organ failure. In New World Leishmaniasis, cutaneous is caused by Leishmania brasiliensis, Leishmania amazonensis, and L. mexicana. And then the visceral is caused by L. chagassi, which is not associated with Chagas disease, by the way, but is caused by L. chagassi. And then we have the Old World Leishmaniasis, both cutaneous and visceral. I'm sure you can read the slide here on the different species, but we're going to be concerned mostly about the diseases. So Leishmaniasis in vertebrates is a pretty nasty disease. You can see I got a lot of pictures here, so we'll go over them in a minute. But one thing is they are what are known as intracellular obligates, and they are found in the macrophages of vertebrates. So these protists gain entry into the white blood cells, the macrophages in particular, and they enter in there for replication. Now, we have a system called the reticuloendothelial system, and these are monocytes and macrophages that are in tissue. So reticuloendothelial simply just means within tissue and within endothelial cells. And these white cells move around. That's what these monocytes and macrophages do. Monocytes are usually found in circulation and macrophages are found in tissue. They are most commonly found in the lymph tissue, in the spleen, and in the liver. When a white blood cell, when a macrophage is infected, right here we have a macrophage, when it's infected with the actual Leishmania parasite, sorry about that, it creates what are called Leishman-Donovan bodies. So here we have some amastogotes here, right? These guys are all outside. Here is the nucleus of our macrophage in the cytoplasm, and you can see all these little dots here we have. These are amastogotes, and so these are infecting and getting inside of these white blood cells. Here we have more. So here's more amastogotes, top right. Right, so we have these amastogotes here and here. This, of course, is the nucleus. More here, right? My pad does not want to cooperate here. So here we have nuclei. These guys are fusing, and we have all these amastogotes here. Same thing's going on here where they're pointing the arrows. Those are the amastogotes inside in these white blood cells, and you can see these nuclei. So when they form these Leishmania-Donovan bodies, they are in their amastogote form. They reproduce through binary fission, so they're going to start dividing. A single cell can contain a couple hundred Leishman-Donovan bodies, and once these once the parasite increases to a certain number, you can see this guy here, he's got quite a few in here. Eventually the cell's going to rupture open. And when the cell ruptures open, all of these amastogotes are going to be released out into the extracellular space to be engulfed by another white blood cell. Think about back when we did the immunology lecture in class, we talked about macrophages in particular, because macrophages are highly phagocytic cells. They are very phagocytic, and they're going to recognize this parasite. It's very small in this amastogotic form, and it's going to be taken up. It's going to be phagocytized by the macrophage. And once it gets inside of the macrophage, it'll start this process again. It'll go into binary fission, build up to a couple of hundred Leishman-Donovan bodies, rupture the cell, and then it starts all over again. Now, Leishmania in the sandfly behaves a little bit differently. So in human cells, it's in the amastogote form, but in the sandfly, it's in the pro and paramastogote form. So it starts out, the sandfly will ingest a blood meal, and so it takes in a blood meal from a human host, and the human is infected with Leishmania. The macrophages and monocytes are infected with the amastogotic form of the Leishmania parasite. Once in the hindgut, the amastogotes will start to transform, and they're going to change into first a promastogote. So that's what we've got over here. We have promastogotes here. They're very elongated, they're pear-shaped, and their kinetoplast is lateral to the nucleus. So here is the kinetoplast down here, and here we have the nucleus. So they are lateral to each other. Here's our promastogote here. Okay, this would be a promastogote, and then we have the paramastogote over here. Once the promastogote has elongated and it's going to be lateral to the nucleus, it then develops into the paramastogote. And the paramastogote is very round and more oval shaped, and its kinetoplast is going to be below the nucleus. So here we have a promastogote. You can see they're lateral here, but here in the paramastogote, this is the nucleus. It's below it. I know it looks like it's above here, but it's closer to the flagella. So it's below it. Here we have a promastogote, and here we have a paramastogote. After the amastogote transforms into a pro or paramastogote, it's going to move its way up into the foregut. So it develops into a pro or paramastogote in the hindgut, then migrates up into the foregut and becomes metacyclic. A metacyclic promastogote is the infective form, and it's going to move into the proboscis and mouth parts of the sandfly, so that when this sandfly moves on to take another blood meal, it will first, of course, inject saliva as an anticoagulant, And in that saliva will be the metacyclic form or metacyclic form of the promastigote. Leishmaniasis distribution, we have visceral cutaneous and mucocutaneous. So green here is where we have most of the visceral, right? And that's, remember, this starts out with lesions and such and then becomes systemic. So we have in the green here. And then around here, cutaneous and subcutaneous. You can see we have lots of it over here in the Middle East and up into India and such. Now over here in the old world, we've got the same thing going on, right? We have visceral on the outer edges, but most of it is mucocutaneous or cutaneous. So a lot of it in Central and South America. Look at that. We have some up here in Texas, right? So right here in Texas. Now, leishmaniasis is transmitted through a 3-component cycle. Let's start with the sandfly stages, right? So we have our sandfly stages here, bottom left, and sandfly takes a blood meal. We're going to start here, right? So here's one. Sandfly takes a blood meal, ingests macrophages that are found within that blood, and those macrophages are infected with amastogotes. Now remember, once they get ingested, they're going to move into the hindgut. Amastogotes are going to transform into promastigotes into the midgut. And of course the paramastigotes. They'll start dividing within the midgut, move up into the proboscis of the actual sandfly itself, and the sandfly, this new sandfly, is now going to blood feed. But this guy, if he blood feeds on a human, then we have, of course, the human stages, right, where promastigotes develop into amastogotes inside of our macrophages, and it starts the cycle all over again. But what we have, particularly in old world leishmaniasis, is we have this third component here, and that's dogs. There are lots and lots and lots of stray dogs, particularly in the Middle East. And this is very much a cultural thing. Now, these dogs are usually kept in order to guard these small rural villages. The villagers will feed them and give them food, but they do not befriend them. They're not pets. The dogs live on the outskirts of these village areas. They know there's food there, so they stick around. And the people keep the dogs there, and it's kind of this interesting mammalian type symbiotic relationship where the dogs will guard and they will warn people if something is coming or happening because they want to protect their food source. So the humans semi-domesticate them as a type of alarm or a type of warning system and protection, and the dogs, of course, in turn are provided meals. So anyway, these dogs, there's lots of dogs, and if you have a bunch of people in a village that have leishmaniasis, they're going to these sandflies here are going to feed on the dogs as well as feeding on the humans. And very much the same thing happens in the dog as happens in the humans. So the promastigotes get phagocytized by macrophages in the dogs, develop into amastogotes, bust out, right, to get taken up by more, so the infection spreads. But at some point in time, that mosquito takes a blood meal off the dog. And it can actually transmit from dog to human and from human to dog. So we could say that this was a epizootic type of disease here. Now, the life cycle itself, we're just going to look at the 2 components between humans and sandflies. And I have a couple pictures here of each. So here we are in our para and promastigotic form, right? Look, we have 2 little nuclei in this one, the kinetoplastids. The sandfly takes that blood meal. Over here in the human stages, here we have all of these amastogotes inside of this macrophage. And down here is a really great picture of the macrophage busting open. And look at all of these amastogotes are all spreading out into the extracellular area. So they move out into that extracellular area. They get taken up by new macrophages. And at some point in time, this person is going to be bit by another mosquito, and that mosquito takes in that blood meal. Amastogotes, right? They move up into the midgut. They're in the midgut, become promastigotes, paramastigotes, and eventually move up into the proboscis to be spread back into the human again. So when sandflies are blood feeding, they inject the promastigotic stage into the human. And the promastigote is going to be phagocytized. Once it gets into the actual macrophage itself is when it changes to an amastigote. This is known as the tissue stage in what we look for in a blood smear as a Leishman body. We look specifically for macrophages in a blood smear and look inside those macrophages. If the macrophages have all of those little stained dots, that's done with staining, have all of those little purple dots, then and based on some patient history, we can determine whether we're looking at amastigotes or Leishman bodies in macrophages. This disease can spread pretty quickly because they can replicate fairly well. And bust open, and a single parasite in a macrophage can develop into 200 or 300 parasites, which can then burst out into the extracellular space and be phagocytized again. Leishmania species and other factors determine if the infection is going to be cutaneous, mucocutaneous, and visceral. So different Leishmaniasis species can cause different forms of the disease. The sandfly that is feeding off of this infected person is going to take a meal that has infected macrophages. We know amastigotes in the sandfly turn into the promastigotes in the hindgut or midgut. This too is going to be species specific. So some sandfly amastigotes transform in the hindgut, others do it in the midgut. Eventually they're going to migrate to the proboscis and the new host will be infected. So let's take a look at some of the differences in these diseases. Cutaneous Leishmaniasis is sometimes referred to as dermal, and where the sandfly bites is where the infection begins. It usually starts out as a macule, develops into a nodule. It can ulcerate, and then if it heals over time, it will cause some pretty nasty scarring. This can take a while to develop. The ulcerative form is the one form, right? We have an ulcerative form right here. This is the one form that we're used to seeing, and it usually does leave behind some pretty nasty scars. This can result from a single bite or lots of bites, and it can travel through metastasis in lymph. So what that means, if you've ever heard of metastatic cancer, what this means is when the parasite gets into this wound here, right? So this person got bit by a sandfly and they have developed cutaneous leishmaniasis. But right now the disease is kind of isolated to this area on the foot. It's not really going anywhere. But if left untreated, it can actually move through a process known as metastasis. In metastasis here, the monocytes get infected. Remember, I said macrophages are found in tissue, but monocytes are found in blood. And macrophages ultimately come from monocytes. Monocytes are the immature form of a macrophage. So when a infection like this occurs, monocytes and macrophages are responding to this site. And the macrophages, they're going to be in tissue. So that's what's infected in this area. But if monocytes begin to get infected by this, then the monocytes are found in the bloodstream, and metastasis occurs when we have movement of a disease from one area to another. So in this case, what started as just simple cutaneous leishmaniasis can quickly become systemic or become visceral. Cutaneous leishmania is the most common form. It's the one you see the most often. New World reservoirs. There are multiple warm-blooded mammals and marsupials that are part of the life cycle, or we could say the reservoir. So in the New World, reservoirs include, we have a whole host of organisms here, right? Humans and dogs are not the only ones. Anteaters, armadillos, sloths, raccoons, horses, possums, monkeys. We have all kinds of different hosts. Old World reservoirs include the monkeys, rodents, dogs, and hyraxes. Hyraxes. These are a wild animal often used as a food source. Now, the big problem, like I was saying earlier, are the stray dog populations. These stray dog populations are in close proximity to humans, and so the transfer of the disease back and forth is a big issue. One way that they have been fighting this is they haven't been able to come up with a vaccine against leishmaniasis in humans, but they have come up with some that are fairly efficient in dogs. So by reducing the Leishmania population in dogs, they are now able to decrease the incidence of Leishmania in humans. Mucocutaneous Leishmaniasis is pretty rough stuff. It causes nasal, mouth, and mucous membrane lesions and ulcers. It's called mucocutaneous because of where it's occurring, so it'll start to heal, and then after it heals, you start getting like cutaneous. You'll get an ulcer from a cutaneous leishmania, and then all of a sudden it'll come back. So inside the mouth, inside the nasal cavities, it's mostly involving the mucous membranes around the face. Eventually, it will consume and eat away. Look at this. I mean, it just eats away at the cartilage. It just tears up and breaks down all of this soft tissue in there. Leishmaniasis, mucocutaneous leishmaniasis in the nose and loss of the septum in the nose is very common in this form of the disease. Visceral Leishmaniasis has a couple of different names. It's called Kala Azar and dum-dum fever. It's a chronic systemic form of leishmaniasis, and it's pretty nasty stuff. It usually starts as a very small lesion, and it becomes metastatic quite quickly. The parasite will pop out of when a macrophage busts open. The parasite will actually start to travel through the bloodstream. And when it starts moving through the bloodstream, we now have a systemic type infection, and it causes fever, enlarged lymph nodes, what's known as hepatomegaly, which is enlargement of the liver, and splenomegaly, which is enlargement of the spleen. That's what you're seeing right here. Okay, and this young child here, usually that's hepatomegaly. Now, that's not all entirely liver, but the liver and spleen are enlarging, putting pressure in the abdominal area here, and a lot of fluid buildup from inflammation. There will also be a decrease in the red blood cell count, the RBCs. The patient will become anemic. It is has a pretty obvious clinical presentation. You see these like real divoted type ulcers. Here we have what is thought to be maybe a mucocutaneous, but this person has probably spread into cutaneous. This right here, the hepata and splenomegaly, are both very obvious signs. This gentleman here, I'm not sure. This is a pretty extreme case, but we have all kinds of different types of lesions occurring here. It has about a 2 to 4 month incubation period. So once it gets into the body, it can take a long time for it to actually start manifesting. Sometimes lesions, right here, cutaneous lesions appear later and can last up to 20 years. These are not easy to get rid of. They're very painful and very difficult to get rid of. There are a few different treatments here in the United States. If particularly soldiers may have been bitten by sandflies, if they believe they're developing leishmaniasis or at risk, they'll give them IV administration of a drug known as pentastam. In 2014, the CDC introduced miltefosine. It's not approved for women who are pregnant, so if you're pregnant, you cannot use this particular drug. Parasitic diseases are a little tough to treat sometimes. What type of treatment a patient is going to have is going to be determined by, of course, their own health, their current health, that's their patient history, but also what species of Leishmania are they infected with, how far along in the infection are they, how many lesions do they have. Treatment for Leishmaniasis is extremely customized because of the number of different species and conditions, and of course the 3 different types of infection. Is this a systemic? Where is it going? Or is it just a localized region? So the treatment is very customized, and it can be very expensive. Other diseases that are caused by Phlebotominae flies is vesicular stomatis virus, right, VSV. That is a virus that is related to rabies. It's known as a rhabdovirus. That's the family of viruses. It occurs mostly in livestock, horses in particular. It's very rarely found in humans, but in horses it's a big deal. And in parts of Central and South America, horses are used quite a bit in farming, so it's important. Humans can contract it through contact with the fluid from animals. So you can see here, and these are horses, we have a lot of saliva, this crusting around the mouth, and it's very similar to rabies at first clinical representation. Excessive salivation, fear of water, open sores. In pigs, when it's found in pigs, they'll have lesions on their feet. They get these like blister-like type structures. Sandfly fever is known as bunyavirus or Papatasi fever and Phlebotomus fever. Sorry about that. And there are 5 different types or serotypes of this bunyavirus known as sandfly fever. In Central and South America, it's called Shandiru, Chagres, and Punta Toro. These are what we call serotypes or subspecies, 5 different viral bunyavirus species. In Southern Europe and Africa, China, Naples, and Sicilian strains, and these are, sandfly fever is pretty common. It's non-lethal, but it's not a lot of fun, kind of like a really nasty or a very mild almost malaria. It is sometimes mistaken or thought this person may have dengue or yellow fever because of the joint pain. There is joint pain associated with it, but there's usually some kind of joint pain with a fever anyway. It is a zoonotic disease, so it easily moves back and forth between humans and animals. The Changuinola virus is a reovirus. Reovirus is the family. It's found in Brazil, Panama, Colombia, and it's called transovarial transmission. So it's transmitted usually through the eggs or larvae. Most infections occur in young children. They're pretty asymptomatic. It's a very mild disease, non-lethal, and most of the time people don't even know that they have it. Bartonellosis. This is known as Carrions Disease. Here you can see the Bartonella bacteria inside of these cells here, in these red blood cells. It's sometimes referred to as Verruga peruana and Arroyo fever. These are gram-negative rods that have motility. They are flagellated. So Bartonella bacilliformis is the name of the bacteria. It is a gram-positive, it is a gram-negative rod. The bacteria grows within. It is an intracellular obligate to red blood cells. It can also be found in the cytoplasm of endothelial cells. Now, Bartonella, other different species of Bartonella bacteria cause other diseases, including trench fever, which we're going to look at a little bit later, and cat scratch fever. But neither one of those diseases are associated with sandflies. Now, cat scratch fever, Bartonella of certain species grows prolifically in the saliva of cats. So cats, and it's normal flora for them. So when cats clean themselves, in particular their claws, they'll get this Bartonella on their claws, and when people get scratched, the Bartonella will actually get in the wound and then cause disease. Bartonella from cats infects the lymph nodes. Now, Bartonellosis in humans presents in 2 different ways. We have Verruga peruana, which are these kind of skin eruptions, like these skin lesions. That's what we have here. Pretty mild. It's sometimes referred to as a wart, the Peruvian wart. It can occur alone, or it can occur with what's known as Arroyo fever. Arroyo fever has a really high mortality rate, about 90% fatality. So it's a really nasty disease. Causes enlarged lymph nodes, severe anemia, severe muscle and joint pain, high fevers, headaches, and it's sometimes referred to as febrile anemia. And that's what we're seeing up here. Occurs primarily, again, in children. Now, Bartonella is oftentimes diagnosed or seen in what's known as a thin blood smear. And this is important because many of the areas that are infected by, or many of the areas in which people are infected with Bartonella are actually quite rural. And in these rural areas, they don't necessarily always have real fancy equipment around to do antibody testing or anything like that. So some places do, however, at least have a microscope and the ability to take a look at these organisms under the microscope. So here we have a blood smear. It's found in a thin blood smear, and the Bartonella itself, you can see right here where the arrows are actually attached. Sorry. They are actually attached to the outside of the red blood cell. If you just look at where the arrows are, you can see it attached to those red blood cells. Once it gets inside, so this is early stage, right? So up here we have early stage infection. Down here at the bottom we have a late stage infection. So later on, once they've gained entry into these red blood cells, look at this cell here that the arrow is pointing at. This cell down here at the bottom, it really has just a bunch of bacteria inside of it. At this stage, red blood cells are not functioning properly and the patient has severe anemia. Up here in the top right, this is a gram stain of Bartonella. So gram-negative bacteria will stain pink, which you see here, right? So these are all, not this whole cell, this whole red area, this is millions of bacteria right there, right? So here we have a single one. Here's a couple of cells, a couple of cells. Those are all Bartonella. Those are gram-negative bacteria. Over here, where we see these purple dots, those are gram-positive bacteria. They are not what we are looking for. Bartonellosis is treated with antibiotics. Cipro is the most common and preferred method of treatment, but other classes of antibiotics that work are the rifamycins, chloramphenicals, and aminoglycosides. Some of the common names, right, are chloramphenicol, most people have heard of. Rifampin is not, that's a commercial name, it's not as common. Gentamicin is a pretty common one. Now, Bartonella does have some of what we call beta-lactam resistance, and beta-lactam resistance is associated with penicillin resistance. Beta-lactam rings are a specific structure in these classes of drugs, and organisms such as Bartonella that are resistant to this drug are so because they cut that beta-lactam ring within that molecule, and the drug can no longer function. So if beta-lactam resistance is thought to be occurring, then they're going to use different drugs in combination with chloramphenicol. So they'll start giving multi-antibiotics together. So that's the end of the Sandfly lecture. I hope you got a little bit of something out of it and didn't gross you out too much. And I will see you all in class. Have a great night.